Okay, we're going to move into section 8.2 and hopefully finish this up. We're going to talk about disaccharides. As I left off in the last lecture, I talked about how you can use the alcohol on one sugar to form a glycosidic bond to another sugar. And so something that we need to be able to do, a skill to develop, is actually be able to identify that linkage. And so how we identify the linkage is if we draw the Haworth projection in the standard form with this oxygen up here in the top right, then we read this from left to right. We go from anomeric carbon to the alcohol on the other sugar. As I talked about, this bond right here is fixed, and this green one is variable in nature. So in fact, we can see on this example over here that it is now in the beta configuration, but notice that this oxygen that I'm highlighting in orange is still down. And so how we indicate the type of bond is we indicate the anomer and which carbons are being linked. So this is an alpha 1,4 linkage on the left and a beta 1,4 linkage on the right. And so in disaccharides, you'll link from the anomeric carbon to some other oxygen. Now, sometimes they're hard to draw, and so there's another way to do that. And there's variations on this. Sometimes you'll see curvy lines, which I think are actually a little bit better than what's being shown here, where they actually have these bends, because hopefully, as we were taught in organic chemistry, each bend identifies each corner identifies a bond. Now, the reason they do this is it just ends up being a little bit easier to align the sugars. And so how you identify their anomeric nature is you look at the relative direction that they're going. And so in this case here, we're coming off the anomeric carbon. I know it's the anomeric carbon because it is the carbon that is shared by two oxygens. So that's always one way to identify the anomeric carbon. This is a D sugar that is pointed down, so that's an alpha linkage. Now, this is an interesting disaccharide in the fact that it actually is linking anomeric carbon to anomeric carbon. And again, how I can identify that that is an anomeric carbon is it's an oxygen to a carbon to oxygen bond there. So this is actually what's referred to as an alpha 1. So we're in the alpha configuration on from carbon 1 to a beta 2 linkage. Alpha 1 to beta. Okay, we'll talk a little bit more about sucrose in a second. Again, you can see here lactose, and like I said, the main thing here is just understanding that there's a couple different ways to show these linkages. Let me just go back. If you try to actually align them in space how they should be, sometimes you get this nice one when you have an alpha linkage here where they just kind of run in a straight line. If you get this beta linkage here, you start to get these kinks, and it actually depends on which oxygen you're using. So sometimes these can get cumbersome to draw. So they use these little squiggly lines that we'll see more examples of to help us understand. But the key is that there's only one bond in between the anomeric carbon and that oxygen. So just a bond between that carbon and that oxygen there. And this indicates that the oxygen's coming out from the bottom, and this indicates the oxygen's coming out from the top. So a couple interesting disaccharides is we've got sucrose and lactose and maltose. And so let's dive in and talk a little bit more about those. So you can get a variety of different types of disaccharides because you can link a bunch of different sugars. And you can link those sugars with different anomers, and you can link them to a variety of different OH groups on there. So we're going to talk about maltose and lactose and sucrose, and you can see that for maltose, it's just two glucose units hooked together. Lactose, we change up one sugar and we get a galactose and a glucose, and then you get glucose and fructose. And so anyway, take home messages, we can get a lot of variety here because you get different anomers bonding and you can use different hydroxy groups to make that glycoside bond. So when we look at naming, don't worry, we're not going to have any naming here, but you should be able to parse the name. And what I mean by that is take information from the name to help you identify what the structure is. So you might not have to provide a name, but you should be able to take from this name that we're using a glucose and a fructose. That glucose is hooked using carbon 1 to carbon 2 of the fructose. The glucose is in the alpha anomer and the fructose is in the beta anomer. And we're linking them by oxygens. So that's what this is. So it's an oxygen linking those two sugars together. So we'll see some N1s in the future. For the first monosaccharide, instead of calling it a pyranose, we call it a pyranosyl, YL, kind of like a methyl or an ethyl, meaning it is part of a larger molecule or substituent. The arrow is showing the connection from left to right. And anyway, so that's the naming. Like I said, we won't have to name them, but you should be able to parse what the name means. So here we've got an oxygen linkage between a glucose and a glucose, between position 1 and position 4, and the anomeric configuration is alpha. So maltose, also known as malt sugar, is actually prepared by breaking down a starch. It's two glucose units linked together. It has an alpha linkage, and we can actually break this down so you can digest malt sugar. So if you like Whoppers or malts, this is one of the components of that, or it used to be. And so we can actually take this out, or sorry, digest this. There is a reducing end here, and what we mean by a reducing end is there is a carbon that's in the hemiacetal form that could open up to an aldehyde and be oxidized. We'll contrast that in a second with sucrose that is not a reducing sugar. Here's lactose. The difference between it is twofold. It is a beta linkage as well as containing galactose. The difference between galactose and glucose is galactose has the OH up here. Now this unit to the right is actually glucose, so you can see at position 4 that OH is down for glucose. So again, those in green are fixed. The position that I'm highlighting in orange is variable. Those are the anomeric positions. And so in lactose, it is a beta 1,4 linkage. And most people can digest this, although as you get older, you may get lactose intolerant because mammals aren't necessarily meant to be fed milk their whole life. So for a lot of people, the genes to digest lactose or milk sugar actually are turned off over time. And so what happens is as you drink milk or dairy products, the lactose goes into your intestine, and the bacteria there decide that they will make something out of it, and that tends to be a lot of gas and acids, and it kind of ferments in your stomach and yeah, it makes you feel uncomfortable. So that is lactose, a link between galactose and glucose, and it's an example of a beta 1,4 linkage. Here is another example of a beta 1,4 linkage. Now what's interesting is the previous one, lactose, which has a glucose, and we can digest it, or most people can. Here we've actually got two glucose units. So I'm gonna draw in blue here, put some parentheses around that, and call that galactose. So the difference between galactose and this cellobiose, or sorry, let me back up on that. The difference between lactose and cellobiose is simply lactose contains a galactose, which I've indicated here in blue, and cellobiose has two glucose units hooked together by a beta 1,4 linkage. Now, this is the disaccharide of cellulose, which most mammals cannot digest. And so even cows and termites can't digest. It's the bacteria in their stomach that can digest it. So if you just chew on some grass, it will pass right through you. But if cows do, it's the bacteria in their stomachs. These are called ruminants. So they ruminate on the cellulose, and the bacteria eventually break it down and release the glucose units. So it's interesting that we can break down, or most people can, a beta 1,4 linkage between galactose and glucose, but humans don't have the enzyme to break down a glucose-glucose beta 1,4 glycosidic linkage. Okay, we're going to see more of cellobiose. That's just kind of the disaccharide of cellulose. So we'll talk about cellulose a little bit more in a second. So sucrose is a really interesting sugar because you connect anomeric carbons to anomeric carbons. So you connect the alpha anomer of glucose to the beta anomer of fructose. One way to tell that you have a ketose sugar that I haven't stressed before is you look at the anomeric carbon, and the question is, how do I know that's the anomeric carbon? Well, like always, it's the oxygen connected to a carbon connected to an oxygen. That is the anomeric carbon. This is an oxygen connected to a carbon. That is an oxygen. And so both of these in orange are the anomeric carbons. And so you're connecting the anomeric carbons together. There is not a hemiacetal present here, so this is a non-reducing sugar. This doesn't open up and undergo mutarotation. This is fairly locked together. And as I was saying, you can identify that you actually have a ketose sugar, because if I look at the anomeric carbon, notice I've got a carbon off of that anomeric carbon. That means that in a previous life, this group that I'm highlighting in red was a ketone. Now here, there is a hydrogen off of that carbon. So this group right here in its previous life, or the open chain form, is actually a aldehyde. So that's an aldose, and fructose is a ketose. So you can tell those type of sugars just by looking at the anomeric carbon. If you have an H off of there, okay, this came from an aldose sugar. If you have a carbon off of that anomeric position, this came from a ketose sugar. So something else to watch out for. So that's sucrose. Interesting because it links these anomeric carbons together. It's also known as table sugar. So pure sucrose or table sugar is a disaccharide. We can hydrolyze it. You can break it open, and then you get glucose and fructose out of it. But as table sugar, it is a non-reducing sugar. It doesn't undergo mutarotation because it's not a hemiacetal. Okay, as a little side note, there's a box in there that talks about artificial sweeteners. So people have tried to do away with sugars because they taste good and have calories. And so if you're like me, you live off the 3 food groups, which is sugar, fats, and chocolate. So if you can reduce your sugar intake, you're probably going to be more healthy. And so one way to do that is to get a non-nutritive sweetener, something that tastes sweet, but your body doesn't digest and therefore get calories from. And another way is to have a compound that you can use in much smaller amounts and still taste sweet. So these are all things that artificial sweeteners do. So they're either non-nutritive, or they're much, much sweeter than normal sweetening agents, such as sucrose, which we can actually see here is used as the basis for this test. And so fructose is actually a little bit sweeter. And then you have all these synthetic molecules that you can see in the box. And if you notice, they're not very similar at all. In fact, this is a dipeptide right here. You got some complex molecules. This one looks most like a sugar. In fact, it is just sucrose that has 3 chlorines substituted on it, known as sucralose. Sucralose is actually much sweeter than sucrose, about 600 times. So you can use a lot less of it. Your body probably still digests it, but oh wait, I take that back. It's eliminated, unmetabolized. But you can use 600 times less of it. So yeah, for some reason, putting those chlorines on there, it just passes right through you. But one advantage is you can actually use a lot less of it. This one here actually is would add calories because it's essentially a dipeptide, a protein, but it has a much higher sweetness than sucrose. So again, you can use a lot less of it. So just appreciate that there's synthetic molecules out there. One interesting one that I found out is sweet, but you probably don't want to try, is dichloromethane. Playing with it in the lab one time, it splashed in my mouth, and I'm like, that's sweet, but I would recommend that you don't use that as an artificial sweetener. It's got a terrible side effect of causing cancer. Here's Neotame, that's actually 1,000,000 times sweeter than sucrose. So in this case, note this, sorry, I take that back, not 1,000,000 times, it would be 10,000 times sweeter. In this case, it's a different scale, so sucrose is 100 instead of the previous one at 1. So I just got these from different places. Again, don't worry about the numbers, just understand that we can come up with molecules that just happen to bind sensors on our tongue and give us the sense of sweetness. And don't ever mistake when your boss tells you to go test something with taste. If you're wondering what that means, make sure you go and read box 8-2. Okay, we're moving beyond disaccharides into polysaccharides, and you can classify the polysaccharides in a couple different ways, whether they're branched or unbranched, and whether they're homopolysaccharides, meaning they're all composed of the same monosaccharide unit, or if they're heteropolysaccharides, where they have multiple different units. So a couple different ways to categorize them. What we'll kind of focus on is looking at the type of monosaccharides that are included in these polysaccharides, the anomeric forms that they're found in, and what those linkages are. So one of the first ones that we'll look at is just this polymer of glucose with an alpha-1 4-glycosidic linkage. And this is known as starch, or more particularly amylose. So starch is actually composed of 2 different components, one amylose and one amylose pectin that we'll talk about in just a second. Amylose pectin tends to be branched, where amylose is just a straight chain. So again, this alpha-1 4 linkage is something that we can break down. But the beta-1 4 linkage between glucose units, we can't. So that is starch. It's the main component of flour, potatoes, rice, beans. So this is how plants actually store their glucose. Now, related to starch is cellulose. And with cellulose, it has a beta-1 4 linkage. And we'll come back to cellulose in a second. I just wanted to show you the differences in the structures. Starch ends up forming kind of a helical structure due to that alpha-1 4 linkage, where the beta-1 4 linkage actually favors a straight chain. So you'll find out with cellulose, you get fibers and sheets, where starch and also glycogen that we'll talk about, you end up getting kind of these helical type structures. In fact, let's look a little bit more at starch. So again, this is just the linear form of starch amylose. It's got this alpha-1 4 linkage, and it just happens that with that stereochemistry there, you get a coil, or kind of a helice, If you've ever done the test for starch, you use iodine and it turns purple. You often do this in chemistry labs or even in biology. And what's interesting is the iodine actually forms a complex within the interior of that helix, and that's what gives rise to the dark blue color. If you throw an enzyme in that chews up and breaks down these bonds just to their individual structures, that blue color will go away. It's this helical nature that nestles that iodine in there that gives the deep blue color. So you can either test for starch with iodine or test for iodine with starch. Now, like I said, starch is composed of two things, the amylose, which is the linear form, and the amylopectin. Now, amylopectin has also the alpha-1,4 linkage, and you can see those here. But every so often, about every 25 glucose residues, you'll branch off of the 6th position there. So this is overestimating the branching, but yeah, so you should have about 25 of the sugars before you get a branch there. It's just showing you that how you can get branches here. So in starch, it's branched, but not very frequently. And in fact, here's a better picture of that. So you got amylopectin down here that actually has branching about every 25 residues. Now there's a related molecule known as glycogen that actually branches about every 10 residues. And so it has many more branch points. Now glycogen is how humans store our glucose, and this is how plants store it. And so one of the reasons that humans want to store their glucose in highly branched form is, well, you don't want a bunch of free glucose because you get huge osmotic pressures. So you store it as a single molecule. And what you want is a bunch of ends. So when you need to release that glucose, you can sick the enzymes on it and you have a lot of endpoints to release that. If I was in class, I would scream at you really loud right now, and you would experience a rush of adrenaline and an increase of glucose into your blood. It's called the fight or flight response. And what happens is all of a sudden enzymes start cleaving off the glucose from the ends of your glycogen, drop that into your blood, give you a rush of energy, and that helps you get away from predators or a threat. Now, trees don't necessarily need to get up and run very frequently, so it takes a lot of energy to add these branches in and to break them down. So they put those in less frequently. You'd still like the branches instead of just a linear part because, you know, when you do need to release the glucose, you will have multiple ends. If this was just a linear, then really you could only cleave it from the ends. As long as it was an exoglycosidase, something that's cleaving from the ends there. Now, if you read in your book, you'll talk about how we start to digest starch within our mouth using an enzyme that will just non-specifically start to break apart the starch there. Okay, so glycogen, we store it. It is highly branched and it has the alpha-1,4 linkage as well as the 1,6 linkages that give it the branching at a much higher rate than we see in the amylopectin. Now cellulose has the beta-1,4 glycosidic linkage. And as we talked about before, that makes it nicer to draw and also means that the cellulose tends to be more in a fiber form. And these fibers tend to form large aggregate structures that are held together by hydrogen bonding. And so cellulose is the main component of wood and plant fiber. So you can see the two structures here, amylose, where you got that alpha-1,4 linkage, and it looks like it's stepping down, but really what it's going to do is end up forming a coil, where the beta-1,4 linkage, it just runs in a straight line there. So we'll look at the structures. So again, amylose will coil, and cellulose, you get lots of hydrogen bonding between the strands that make those cellulose molecules more into fibers and sheets. Cellulose, even though it has a bunch of OHs, tends to be very water insoluble, and that's due to the extensive hydrogen bond nature that it has between molecules. And also, you notice all those OHs are pointed outward, so you do kind of get this hydrophobic base right here. And so the cellulose sheets will stack together and kind of take advantage of that hydrophobic interaction there. Okay, we need to talk about some modified sugars. So this is called chitin, and I'm not kiting you. Some people pronounce it different, as if there were an S there, but I won't say it because this is going on YouTube and I don't want my mom to hear it. So chitin is a repeating unit, looks a lot like glucose. It has the beta 1,4 linkage between it, so it would look very similar to cellulose, other than at position 2 there, you have an N-acetyl group. So you've got a nitrogen that's been acetylated, and acetyl group is this thing right here. So this is a biopolymer that is used by insects and other arthropods. It makes up their exoskeleton. You may not know, but we've got a lot of cicadas that are coming out. They come out of the ground every 17 years, I believe. And we're in, I don't know if we're in what they refer to as brood X, but a lot of these guys are crawling out of the ground. And as they eat and grow, they outgrow their skeleton. And so they undergo a process called molting, and they leave behind their exoskeleton. They tend to be vulnerable for a while until they can reestablish a hard exoskeleton, and that exoskeleton, again, is made out of chitin. Likely the second most significant carbohydrate behind cellulose. So, like I said, it's found in all sorts of living things, insects, lobsters, beetles, spiders. It's what gives the lobsters and crabs their hard shell, this chitin. Okay, so we need to talk about some glycosaminoglycans. So these are linear polymers of repeating disaccharide units. And one monomer is either this N-acetylglucosamine that we just looked at before, or N-acetylgalactosamine. They do have one other property besides chitin, and that is they also have a negative charge on them. So they get oxidized at C6, so those are referred to as the uronic acids. And they can also have sulfate esters on them. So these end up forming extended hydrated molecules. Since they've got all these charges, they tend to absorb a lot of water, and these are used to form a meshwork with fibrous proteins to form an extracellular matrix. So this is what's used in your connective tissue and the lubrication of joints. So if you look at these molecules here, you'll see that, yeah, we've got carboxylic acids, we've got these N-acetylglucosamines and the sulfate esters. So there's lots of variation that we can actually get in our carbohydrates, probably more so than you can get in proteins, because they just always link from the N to the C, so they're linear. They may have different side chains, but here you've got a lot more variability. You've got way more than the 4 nucleotide bases, so there's a lot of structural information that can be held in carbohydrates. So here's some examples of the glycosaminoglycans. Again, these form a gel-like matrix that fills extracellular space in the body around collagen and other connective tissues. And you can see the variety of the repeating unit. So you got high uronate there, dermatan sulfate, chondroitin 4-sulfate, keratin. This is different than keratin that we talked about in the protein section. This is keratin sulfate, chondroitin 6-sulfate, and heparin. Notice heparin's got a lot of negative charges on there. So there is a related molecule known as heparan. So we've got these differences between INs and ANs there. There is some guidance on what you need to know in the study guide. The molecules that you need to know are listed there. Take-home messages: carbohydrates can vary quite a bit in their structure. So let's just look at one of these glycosaminoglycans. Oh my goodness, hyaluronate. Oh my goodness, let's spit that out. So hyaluronate is a component of connective tissue. It's found in your synovial fluid, so between your joints, the vitreous humor, and the eye. So if you took the liquid out of your eyeball, you would find this in there. These can be pretty long, 250 to 25,000 disaccharide units per molecule. You can see the repeating unit shown there. These end up having kind of an extended rigid structure. And since they have all these charges on there, like I said, they tend to be highly hydrated. So in fact, when you get all the water molecules there, the volume of this tends to be 1,000 times its dry volume. So it really kind of gels up and expands the volume. It has this viscoelastic behavior. So you get a mess of tangled molecules at low stress. And so as you get more and more stress on that, the molecules tend to start to align and you get less stress or less resistance under shear stress. So it kind of prevents things from sliding until you start putting too much stress on it, and then things will slide but still offer some resistance. So it's not quite like skating on ice, but it's tuned to the amount of stress that you're applying to the two surfaces that are separated by this component. Okay, so some more sulfated glycosaminoglycans. Like I said, we talked about dermatan sulfate, keratin, and heparin. So heparin up here is a cell surface component. It's also found in blood vessels and the brains, and it's critical in development and healing. So that's heparin, but I want to focus a little bit on heparin, which is actually a medication. It's the most highly charged polymer in mammals. It's got a lot of sulfate residues on there. It's stored as granules in mast cells that line arterial walls, and so when those get damaged, you actually release this heparin. And it actually inhibits clotting. Now, clotting is good, but you don't want too much of it. We won't talk about it in this biochemistry, but if you continue on, the clotting is very well regulated, and it's from both ends. So you got promoters of clotting and inhibitors of clotting, and clotting is a very delicate balance. You don't want all of a sudden your blood to just all of a sudden clot up. In fact, it's a catalytic process, and so if you didn't have these inhibitors of clotting, in a short time, all your blood could just coagulate. So heparin is one of those things that they administer to prevent clotting. I've got Dennis Quaid here and his wife. At the time, I believe he has divorced her, and last I knew was dating someone half his age. But they had newborn twins, and they accidentally gave the children, or these twins, 1,000 times the proper dose of the blood thinner. And the problem was the old way, the vials looked very similar. And so Dennis Quaid and his wife at the time were instrumental in getting that changed. And so now there's color codes, and it's a lot more obvious which dose you should give. You can imagine you don't want to give an adult dose to a newborn. That's way more than they need, and so their blood would never clot. And what happens is you get these micro tears in your body all the time. And if your blood never clots, you can essentially bleed out just from little micro tears. So clotting is important. You just need to have it well regulated. Anyway, interesting story about heparin. It is variably sulfated. I believe that they get it from the pigs, or from pigs. But now they have changed it where bacteria will make it, and it's a lot more homogeneous than it used to be. So I'll give you a bonus to talk about and explain the current method of obtaining heparin. So do we still administer heparin from pigs, or is it all generated from bacteria biologically? So I'm not quite sure if we still give it from pigs or not. I think we've moved over, and biotechnically we produce it, and it's a lot more homogeneous and has much more defined responses than it previously used to. Okay, so we will just kind of summarize up some of the structures and roles of polysaccharides. So here's a great summary of different polysaccharides, their repeating unit. So it starts off with starch, but again, starch is composed of amylose and amylose pectin, and you can see the type of branching and how long that branching is. So you can see various different types of polymers there. Plants don't have these glycosaminoglycans, these highly charged species. They tend to have what are referred to as pectins. The residues are often methylated or acetylated. They have a rhamose as kind of a core species there, and then you've got other sugars like arabinose and xylose. So they do have the negative charge there. So you got the galactourinate, which has the carboxylic acid at the C6 position. And so these tend to aggregate in bundles with calcium or other divalent cations between carboxylates. And so if you ever make jams, you'll use the natural pectin from the fruit, as well as likely add some in. And that gets this interconnected network of carbohydrates that gives jam their characteristic texture. So bacteria also take advantage of these carbohydrates in their extracellular matrix, and they excrete hydrated polysaccharides, cellulose-like molecules that are acylated, and these create biofilms. So within these biofilms, you often have a mixture of bacterias, and the type of carbohydrates vary. So here's a picture of Pseudomonas, and it is growing on a petri dish. So it looks like it's almost like a living organism, but it's just on a petri dish there that is covered by a biofilm, and it helps keep the bacteria together and protect them from desiccation. If you ever feel a slimy sensation on your teeth, that is actually the bacteria building a biofilm on your teeth, and they're trying to be protected from you rubbing them off. So these biofilms are kind of excreted, and then kind of like a saran wrap layer for a colony of bacteria. Biofilms can be very bad in the case of objects inserted into our bodies, like catheters. So it's not easy to have like a PICC line or something that's always inserted into your body. What happens is even just normal bacteria will start to colonize that, establish a biofilm, and then pathogenic bacteria can take advantage of that biofilm and get established. Okay, so that is polysaccharides. Let's move on to glycoproteins. And as you can guess, glycoproteins are actually proteins that are connected to carbohydrates. So proteins can contain short or even longer oligosaccharide chains, and they're referred to as glycoproteins. And these carbohydrates are connected either through the oxygens, typically of serine and threonine, or nitrogens in the amide bond of asparagine. And so we'll look at that in more details. So, and they serve all sorts of purposes. One of them is if you actually put them on the outside of a cell, they can actually act as an antifreeze. So for things that are living in salty water that is below freezing, you don't want the water inside you to freeze. And so you get a lot of these carbohydrates on your cell membranes, and that prevents the water from crystallizing, essentially disrupts them so they can't form the ordered hydrogen bonds. So glycoproteins, again, these are proteins bonded to oligosaccharides, and the carbohydrates can make up anywhere from 1 to 90% by weight of the protein. So they can actually be a significant portion of the actual glycoprotein. They're used as cell surface recognition. So in fact, the recognition of cells is often mediated by recognizing the types of carbohydrates on the surface. So infection and prevention of infection is mediated by this. HIV protease inhibitor drugs prevent HIV from recognizing these carbohydrate moieties on the cells and penetrating them. It's involved in fertilization and blood clotting, inflammatory disease. So these glycoproteins are involved in all sorts of things. So proteins that are glycosylated end up being transporters and enzymes and receptors and hormones and structural proteins. So all domains of life make use of these glycoproteins. Now, what's interesting is there's no template for adding the carbohydrates to proteins. So when we make proteins, we have a template. We have the mRNA that we got from the DNA. So what you find is a lot of variability in the carbohydrate content, not only their location, but the types and amounts of those carbohydrates. There's a lot of heterogeneity in these. So let's just start taking a look at one of these glycoproteins. They're known as proteoglycans. So they're covalent and non-covalent aggregation of proteins and glycans, which is just another word for carbohydrates in the extracellular matrix. So again, outside of cells. So the main components, like we talked about, are these hyaluronates. Gosh, for some reason it's easier to call them by their other name, hyaluronic acid. I can say that easier than hyaluronate. And you got these other glycosaminoglycans. So you get a core protein, and then you get these link proteins and smaller oligosaccharides. So you may think that this is a protein, but this is actually the hyaluronate. So it is just a carbohydrate. And then branching off of that carbohydrate, so these black lines are actually the core proteins. And then you got some N-linked oligosaccharides, and then in green, you've got these keratin sulfates and then these chondroitin sulfates there. So they end up in this kind of bottle brush structure. So you got this hyaluronate backbone, and then the bristles have, again, this core protein to which the glycosamine glycans are covalently linked. And they tend to be linked to the serine and threonine residues. So you do have some N-linked oligosaccharides here, but the keratin and chondroitin sulfates tend to be linked to the oxygens of the serine and threonines. So the central strand there ranges from 4,000 to 40,000 angstroms long. Each strand can have 100 associated core proteins. Each binding has around 50 keratin sulfates. So this is actually a very scaled down model. So imagine 50 of those there. And each one of those keratin sulfates is 250 disaccharides long. And then you got about 100 chondroitin sulfates. And those can be up to 1,000. So those should actually be a little bit longer. And actually you'd have quite a few more of the chondroitin sulfates there. These tend to be highly hydrated again, and they're polyanionic, so you've got lots of these carboxylic acids and these sulfate esters on there. And so you get lots of water in there. So they're used, like I said, between the cellular matrix. So cartilage is a meshwork of collagen fibers filled in by these proteoglycans. So one way that cartilage works between your joints is as you put pressure on the joints, you start compressing this, and the water actually is kind of squeezed out of your joints. And then as that pressure is released, then the water comes back in. So it acts as a shock absorber. Now, there is another class, we call these peptidoglycans. These are found in bacteria cell walls. So what a bacteria cell wall is, a rigid walls that give bacteria their shapes, and it also allows them to live in a hypotonic environment, meaning that they can live in an environment that has pure water. And so inside the cell it's going to be very concentrated. So why doesn't water rush into the cell by osmosis and cause the cells to burst? Well, they have a very rigid cell wall around them. And one way to classify bacteria is based on the presence of this cell wall. Well, bacteria mostly have a cell wall, and some have a very thick cell wall. And so when you stain them with Gram stain, that cell wall will take up the stain. And so those are what are referred to as Gram positive, very thick cell walls. And then you've got Gram negative bacteria that have a very thin cell wall. And then they have an outer membrane as well. So they surround that. So it's like an onion. You've got a membrane layer and then this cell wall, and then you got another membrane layer. And so gram-negative bacteria tend to have smaller cell walls, and it's harder for the dye to get past that outer membrane, so they don't take up the Gram stain as well. And having that extra outer membrane actually makes the gram-negative bacteria more resistant to antibiotics. So that's the difference between a gram-positive and a gram-negative. They both have a cell wall. It's just a gram-positive is larger and will take up the Gram stain more readily than the gram-negative bacteria. But if you start reading about it, gram-positive and gram-negative is actually a poor way of classifying bacteria. And there's a lot of crossover. So what is this peptidoglycan structure? So it is a connection between, as you can imagine, peptides and glycans or carbohydrates. And so you get this polysaccharide, you get alternating beta-1,4, and it alternates between the N-acetylglucosamine and the N-acetylmuramic acid. Then connected to that is you have a 3-carbon lactic acid group. And then connected to that lactic acid, you actually have this peptide here. And so you get L-amino acid. This one here is a isolinkage. So here's the amino acid, and you're actually connecting through the carboxylic acid of glutamate, so the side chain there. So this is what's referred to as an isoglutamal linkage there. I believe this is actually a D-amino acid, and then you got lysine, and then you got D-alanine. So even though nature prefers the L-amino acid, bacteria have started incorporating D-amino acids into their cell walls, and that prevents them from being broken down by enzymes that chew up protein, so proteinases. So they can't hydrolyze the D-amino acids because they are used to hydrolyzing L peptide bonds. So you've got this linkage here. So you got a very long linkage of carbohydrates, and then you've got this tag right here. And let's see if we can see how that all fits together. So you got this big long linkage, and off of the N-muramic acid, you've got this linkage of amino acids there. And off of the lysine, there, it connects to a pentaglycine linkage. So you've got glycine that's connected here. And then this is the N-terminus of the glycine that connects to the carboxylic acid of that D-alanine there. So the take-home message from this is you get this very condensed network. So you got these runs of carbohydrates, you got these tails of amino acids, and the linker that cross-links these runs there. Now these end up forming a coil. So this little group right there would be like this, kind of like a fiber, and then that fiber forms coils, and then that coil coils around the bacteria and makes up the cell wall. So you kind of form these coils that end up being like a rope, and those ropes are what make up the cell wall. Now, antibiotics actually inhibit the cell wall, and one of the penicillin, what it does is it inactivates the enzymes that cross-links the strands. So I'm just going to go back, and so when we start cross-linking the strand, if we inhibit that, then really what you're just going to do is have a bunch of these runs here that aren't cross-linked. So you're going to have kind of a leaky cell wall, and so the bacteria can't really grow. If they try to grow, then they just burst, and things can get in, and their insides can get out, and so the bacteria end up dying. And so that is what penicillin does. Unfortunately, bacteria have an enzyme, and it's not one that they made specifically to break down penicillins, but it's one that they have started to optimize to break down penicillins. So from evolutionary pressure, they have what are known as beta-lactamases that come in and break that bond right there. And so bacteria can now break down penicillin. So now what they do is they often give beta-lactamase inhibitors, and I believe clavonic acid is one that breaks down that beta-lactamase, or inhibits that beta-lactamase, and then they'll give you an antibiotic as well. So they kind of have to inhibit the thing that breaks down the antibiotic, and then they actually give you the antibiotic as well. We do have an enzyme, lysozyme, so it's found in tears and mucus and in our other secretions, and it hydrolyzes the beta-1,4 linkage between those carbohydrates. So again, we come back and it would break down this linkage here and actually digest the cell wall, which would be good. So you get bacteria in your mouth and we actually start to digest it so it can't enter into us and cause diseases. So we also glycosylate eukaryotic proteins. We do that at 2 different sites. As I mentioned earlier, we do it at an asparagine, and we actually do it through the amide nitrogen there. And so there is a sequence, and that sequence is we have a serine or a threonine, some amino acid X, and then an asparagine right there. So X is never proline. Remember, proline is just the odd duck, and so it causes kinks and turns in there. And then you rarely get these amino acids at the X position there. You do get O-linked, and we attach them to the amino acids that have O's in their side chains, which are threonine and serine. And this is just an example of the most common saccharide that you would attach there. So less commonly, you would attach galactose or mannose or xylose. Okay. So N-glycosylation occurs at the same time as translation. So remember, we're adding this to an asparagine residue. So as this comes off the ribosome, you attach a oligosaccharide with 9 mannose, 3 glucose, and 2 N-acetylglucosamines. And you can see that depicted schematically here. So that is just attached. And then what you got is you got these glucosidases and mannosidases that actually start trimming up some of these groups here. And so as you trim them, you get down to this core structure right here. And then from this core structure, you start building it up and you add other different sugars on there. So again, this isn't genetically programmed. A lot of this is, I won't say random, but you've got to have the enzymes to do this in the right place. So I'm sure it's regulated somehow, but again, you get a lot of what they refer to as micro heterogeneity among here, because these enzymes aren't following a template. They're just coming along and saying, hey, I want to add a sialic acid here, or hey, let's put a fucose here, or hey, let's put a galactose here. Well, you put a galactose there, now I'm going to put a sialic acid there. So again, hopefully you're seeing that carbohydrate chemistry is pretty complex. Proteins have fairly defined structures with their amino acids. DNA have fairly defined structures with their 4 bases, but carbohydrates just have an incredible amount of complexity due to the different ways in which they can form bonds and actually how they are controlled in the body. So like I said, when you attach this glycoprotein, you get this core structure right here that's in gray that has this N-acetylglucosamine, N-acetylglucosamine, and then you get mannose, mannose, and mannose. And then from this structure in Y here, you get modification. So like I said, you could have a fucose here and, a lot of variability there. So this is kind of the consensus sequence for all these N glycoproteins. So what was I going to say? Anyway, yeah. So O-linked oligosaccharides are linked to the serine and threonine. They really don't have a consensus sequence along here. The asparagine you get serine or threonine, and then you get X, and then you get this asparagine on there. You get a lot less variability, or sorry, a lot more variability on the O-linked. The N-linked, you always have, like I said, this core group down here, that kind of Y group, that we can identify here. N-acetylglucosamine, N-acetylglucosamine, and then this Y of 3 mannoses there. Oh, sorry, I'm gonna make you seasick. But this is this is showing you how the linkage is. So you got a beta 1,4, beta 1,4, alpha 1,3, alpha 1,6 linkage. So that's what these numbers are indicating there. Okay, so these oligosaccharides help determine protein structure and function, so they could help stabilize. But what they find out is a protein can be functional without these oligosaccharides. So they're not always required. So they're often attached to surface loops and turns. So there's a lot to be learned about carbohydrate and these glycoproteins. Do know that from the cell surface, you can get a pretty thick fuzzy layer of these carbohydrates there. And it's interaction between different molecules and this carbohydrate layer that actually mediates a lot of cellular processes. And so with these carbohydrates on the surface, you've got a lot of places to form hydrogen bonds. And so proteins that bind the carbohydrates are known as lectins, and they're ubiquitous and they're very specific. So they bind specific types of carbohydrates on the surface, and they can bind fairly tightly with them. So this picture over here is an interaction between the human galectin-2 and the beta-galactoside. This is just a general picture. We'll get more into this when we talk about signaling in a separate chapter. But you can see all the different ways that oligosaccharides can be attached. They can be attached right to lipids. We'll talk about that in next chapter. They can be attached to transmembrane proteins, and then we have viruses can recognize those on the cell surface, and so they'll recognize a specific cell type and then infect those cells. Toxins can actually recognize those specific carbohydrate signals. Bacteria can recognize them, and we actually even have in our cell membranes proteins that will recognize the carbohydrates on other types of cells. So all sorts of recognition processes are impacted by these carbohydrates. In fact, one of the most interesting one is fertilization of eggs by sperm is mediated by the sperm recognizing the specific carbohydrate sequence on the egg. You may wonder why the sperm just don't go and attack all the cells as they swim. And so they kind of do, but until they find that specific sweet sequence on the egg, they don't drop their DNA load. So I don't know if you've ever seen a movie of a sperm actually fertilizing an egg. It's pretty amazing, all the changes that take place fairly quickly. As soon as that sperm hits its target, it's got to prevent other sperm from entering in. You don't want a bunch of sperm fertilizing the egg multiple times. So anyway, really interesting process. Another one that's very interesting is the blood group antigens. So polysaccharides are centrally involved in cell-cell recognition. So it helps one cell distinguish another cell type. And it is where our blood group antigens come from. So you got the cell surface and you've got an oligosaccharide presented on that surface. So you may see a picture off to the right here where you've got these spikes for the A antigen and the round balls for the B antigen, and then the O blood type doesn't have any of these antigens, and that's actually a lie. So the type A antigen, you notice off to the right is the protein, and the protein is connected to N-acetyl-D-glucosamine, and that is common for A, B, and O. And then you're connected to D-galactose, which has an L-fucose, and so you see in red, all the blood types have that common antigen. So the difference between type A is you have N-acetyl-D-galactosamine, type B has D-galactose, and type O has neither. So you may know type O as the universal donor. And the reason that type O is the universal donor is you can give that to everyone because everyone has that antigen on their blood surface. So in fact, it's rare, blood O, but they're looking for ways to process type A blood and cut that group off, and process type B blood and cut that off. So imagine that they could take all the blood donations and process it, so you just cut off that terminal residue there and you convert all the blood to type O. Then you don't have to worry about who you give it to. Everyone can just take type O. You do have to worry about other factors. So you have to worry about the Rh positive factor. And to be honest, if you look into blood typing, well, last time I talked to someone that worked in a medical lab, they actually test for, I don't know, up to 12 different reactivities. Some are worse than others. The biggest one is if you give type A blood to someone that's type B, your body will recognize this on the blood cells and say, that's not my blood type, and will attack those blood cells. And that ends up being a bad day for you. So that is it. That is our carbohydrates. We'll pick up on lipids next.